I'd welcome Wayne Cutfield, Professor Wayne Cutfield, who's the director of the Better Start National Science Challenge, who's going to talk about another cutting-edge area of obesity research at the moment, bottoms up, the gut microbiome and childhood obesity.
Thank you, Barry. In the next 15 minutes, I'm going to take you through a gallop of a new frontier in health research, our gut microbiome, or gut bugs as I will refer to throughout my presentation. While it might be new to us, it's interesting to note that the Chinese for more than 1,000 years have treated gastroenteritis with the yellow soup, which is stool dissolved in water, not intended to be sipped or savoured, and I'm not sure how effective it was. Now, really in the last 12 to 13 years, there's been an explosion in this field, this new frontier of health research, which is our gut microbiome. And while I'll focus a lot on obesity and a bit on diabetes risk, there's research looking at the impact of the gut microbiome on other areas, such as gut health itself, immunity, behaviour, even autism. So the impact or the effect of the gut microbiome may be much larger than we think. From the publications you can see, not a huge number, but most of those have occurred in the last 5 years, so it really is a growing topical area. There is more to us than we see. How many of you have weighed yourself in the last week? Put your hands up. Okay. The good news is that you're actually 2 kilos lighter than the scales tell you, because you have 2 kilos of passengers, which are your gut bugs. So take 2 kilos off. There are 30 trillion human cells in our body, and there are 30 trillion gut bacterial cells. It's a large number of gut bugs. We think that our stools are all waste. Half of them are actually gut bugs. And that consists of bacteria. Most of the focus is going to be on bacteria, but there are also viruses, fungi, and archaea. Now, 12 or 13 years ago, we had a paradigm, and it was that gut bugs were commensal. In other words, they sat in our bowel and they chomped up waste and survived quite happily, but they didn't actually have any effect on our health and well-being. We now have a different view, a different paradigm, and that is we have a mutualistic relationship with these gut bugs. In other words, there is benefit to them and to us in terms of our health and well-being. When that becomes disordered or dysbiotic, they can actually have an adverse effect on our health and well-being. Now, the story really began with pioneers like Fred Backhead in St. Louis and others, and they did a whole sequence of experiments in mice, and much of what we know about gut bugs has come from mice. Now, mice are not men, but one of the important distinctions between mice and men is that mice are coprophagic. They eat each other's poos. So when you're doing studies of gut bugs and transfer of gut bugs, it's a lot easier to do those in mice, as you might imagine. In this interesting first study from Fred Backhead, you can see that he took germ-free mice. So these are mice that have no bugs, no bacteria in their guts, and they're not exposed to bacteria during their life. So they're the control group, the GF. And then he looked at 2 other groups, the conventional R, and these are germ-free mice that were colonized from birth with gut bugs. And then a group that were colonized just for the last 2 weeks. They're now at adult life. And you can see that the 2 groups exposed to gut bugs were appreciably fatter. And that include epididymal fat, which is one of the fat storage sites in mice. Now, they didn't eat more, they actually ate less. And their metabolic rates were higher. So it appeared that the gut bugs were influencing fat storage. And just to very quickly summarize what he proposed, in fact, was that the gut bugs were influencing fat storage in adipocytes and also in the liver itself through a bunch of adipose regulating factors. Now, we've learnt a lot of other things from human association studies and also studies in mice. If we look at our gut microbiome, there are a number of things that influence it. Diet is a biggie, of course. Just over half of our gut microbiome, which is very dynamic, it changes day by day, is influenced by diet. Lifestyle also has a big effect. Antibiotics, there's nothing quite like nuking the gut microbiome than antibiotics. It can dramatically alter it, usually transiently. alcohol, other drugs, probiotics, illness, stress, physical activity can alter the gut microbiome. But it also appears there is a maintenance population, which is a constant part of the gut microbiome that doesn't change so frequently. Now, if we look within our gut microbiome, our gut bugs, in essence there are, something weird has happened here. Okay, it's slightly jiggered the slides. This is Bacteroides, which is a phyla of bacteria, which is thought generally to be was thought generally to be a healthy bacteria, whereas the baddies were the Firmicutes, and they were much more likely in abundance to lead to increased adiposity, increased insulin resistance, and so on. Now, there's a different paradigm now, and that is that it's not so much about good and bad bacteria or phyla, because there are lots of exceptions to that. It's about diversity. What we need to be healthy is a diverse population. I liken it to a rainforest. A rainforest is going to be much more resilient than, say, a single plantation of pine or totara. It will cope with more environmental stresses if there is greater diversity. And that has been shown in this study comparing humans who either had low bacterial diversity or high bacterial diversity. Those with higher bacterial diversity were more likely to be slimmer, to be less insulin resistant, and which is a risk factor for diabetes, had better lipids, and so on. So there were a number of markers associated with greater diversity. So rather than pick on good and bad bacteria, the focus is increasingly on achieving greater diversity. In terms of the development of the gut microbiome, early life is associated with relatively rapid transition and change. And this reflects breastfeeding through to weaning foods and then to a more adult diet. Now, in this paradigm, recently from Sarah Lynch in the New England Journal of Medicine, she proposes that in fact by the end of childhood the gut microbiome is stable. There are other studies that suggest the gut microbiome is stable from 3 to 4 years of age. Somewhere in here the gut microbiome becomes stable throughout life, and then as we get older we have less diversity present. A really busy slide, and I just want to highlight, and this is really a condensation of dozens and dozens and dozens of studies of just where and how gut bacteria work. The first thing to point out is that gut bacteria have the capacity to scavenge an extra 100 to 200 calories from waste material per day for you. And over time, that's an appreciable increased calorie load. In terms of those with who are obese or have type 2 diabetes, what you find is that the diet and diet effects lead to greater levels of butyrate and other short-chain fatty acids, and this signals a whole cascade of events which you can't easily read, which is reduced insulin sensitivity in muscle, increased lipogenesis in the liver and in adipose tissue, increased inflammatory markers, so things that get into the circulation that promote inflammation, and also an effect in the brain by reducing satiety. So in other words, the effect of gut bugs on our weight and health occurs at multiple different levels. It isn't just shoveling more calories into you, it's doing a whole lot of other things. It's also important to remember, as I've mentioned, that it has an impact on gut health, and I'll come back to that later, immunity and behaviour. Now, there are some unique conditions for childhood in which association studies, so now I'm going to move away from animal models and fundamental physiology to some association studies in humans, and particularly children. And caesarean section, of course, is a very common event, and caesarean section rates in New Zealand approach 25%. So we do have one of the higher caesarean section rates in the world, certainly not the highest, but it's a common event. And there are a number of studies that have shown that caesarean section is associated with childhood obesity. Now, this is a very large study, the largest conducted to date, recently published, in which they looked at children from the GUT study, the Growing Up Today study, an appropriate acronym for this talk. And what they found is that, and they looked at those in the study right through to young adult life, but there was a 25% increased risk of obesity during childhood from those who were exposed to caesarean section. And you might wonder, well, why is that? And of course, if you think about caesarean section, or if you think about delivery, that is the earliest colonisation potentially of the gut microbiome. And for about 4 hours after birth, the pH of the stomach is 7, and gut bugs can get from the mouth to the bowel and colonised. After that time, of course, the pH of the stomach drops, and that's a much more difficult journey for gut bacteria to make safely. If you try and put that into context, because the editorial suggested, well, caesarean 25%, in fact, they looked overall, it was 15%. That doesn't contribute to childhood obesity at all. When you do the maths, we expect about 80 of 1,000 children during childhood to be obese. So if you look at how common caesarean sections occur and 25% risk of obesity, about of 1,000 children, 5 of them could be obese due to caesarean section. Now I say could be because these are association studies. Another interesting association study is early antibiotic exposure. And these studies haven't been replicated in adults, but there are now a number of studies, and again, this is a large recent study, and this group looked at antibiotic exposure in the first year of life and the second year of life. And what they found, which is what a number of studies have found, that children exposed to 3 or more courses of antibiotics in the first year of life and the second year of life have an increased odds ratio of becoming obese. And as I said, similar studies conducted in adults haven't found the same observation, which suggests that there is a sort of critical period in early infancy where children are vulnerable to antibiotic exposure and potentially obesity. Now, there haven't been any intervention studies in which antibiotics have been intentionally given to children to see if they become obese. So remember, these are association studies, but it does raise the question about the liberal use of antibiotics. And while there is growing concern about that with antibiotic resistance, I think we just need to think that there are other issues that could be occurring from early antibiotic exposure in which children may be given inappropriate courses of antibiotics. A third group that I wanted to introduce you to are children born prematurely. About 10% of all children are born prematurely, and we've conducted a number of studies in children born prematurely and shown that by mid to late childhood, they're insulin resistant, again, a risk factor for diabetes, and we've conducted studies in adults born prematurely to show they're even more insulin resistant and they have increased adiposity. Males born prematurely in their mid-40s have an extra 11 kilograms of body fat, and most of that is visceral. So if you think about the early life of children born prematurely, it's a pretty hostile world, particularly for their gut function. So they tend to be fed by parenteral nutrition rather than orally, and I'm talking about the very premature children. They have a lot of high-dose antibiotic exposure. They're exposed to steroids. So there are lots of reasons why their early gut environment may be abnormal. So when we looked at these children late in childhood and compared them to those who are born at term, and we adjusted for everything, activity, diet, ethnicity, socioeconomic status, we found that their gut bacteria were different. So that implied that, and of course that's a cross-sectional study, not a longitudinal study, but it implied that early life events have led to long-term changes in that portion of gut bacteria I said tend to be constant within our GI tracts. We also looked at bacterial activity as well, and we found, so there could be differences in gut bacteria, but how does that relate to the activity of bacteria? Because some of them can be quiescent and some of them can be more active, and there were marked differences in gut bacterial activity. Again, preterm in the green. And interestingly, the major changes in the preterm group were in glutamine and thiamine metabolism, with increased glutamate destruction, and lower glutamate levels have been associated with type 2 diabetes. Now, so I've told you about association studies, and for those of you who are skeptical about the role of gut bacteria, what you will really want to see is, are there intervention studies that actually show that if you manipulate the gut bacteria in humans, you can influence health? And the best data so far relates to Clostridium difficile infection. Now, that's a horrible form of diarrhea which can be life-threatening. Traditionally, it was treated with vancomycin with variable success, 30% less. Now, in this study, and there have been several other studies, and they're all small, what they showed was that if you basically put in a rectal tube, you get a donor, clean up the stool, take the bacteria out and pour it in, then you can have a very high rate of cure for Clostridium difficile infection. And in fact, it is now considered to be one of the optimal forms of therapy for this disorder. Now, I've talked to you about mouse-to-mouse transfer. There are examples of human-to-mouse transfer. So in this somewhat complicated study, they looked at twins who were discordant for BMI, so a fat twin and a thin twin, humans. And they then put those bacteria into germ-free mice. And they found that those given the fat twins bacteria, yes, they got fatter. And then when they mixed that mouse with the fat bacteria with a mouse who had thin bacteria, that mouse got thinner. Similarly, if we look at the mouse exposed to the thin human's gut bacteria, they got thinner, but when you mixed them with the fat mouse, they got a bit fatter. And these changes were more exaggerated in an unhealthy diet. So while it still isn't human to human, it's getting closer. Perhaps the best example is an accidental observation, and it was in Clostridium difficile infection. So the recipient was a woman who'd been slim all of her life. She developed Clostridium difficile infection. The donor was her daughter, who was very overweight. After transfer, this woman became obese and remained obese for years afterwards. So it's the reverse of what we're talking about, which is going from fat to thin, but does suggest, in fact, that there may be a role here in humans. This study in type 2 diabetes, and again, all of these are very small studies. They looked at those with type 2 diabetes, put a nasogastric tube down, put gut bugs into the bowel of those with type 2 diabetes, and found that insulin sensitivity improved considerably by 75% 6 weeks after transplantation. They didn't follow them long enough to look at their BMI and their weight. Let me just step past that because time is on us. So where are we now and what should we be doing in this field? And there is a general consensus that it's time for action. We need to look at intervention studies in humans to see how effective they are. Could this be a meaningful form of therapy beyond all of the association studies we see? So what we're about to get launched on is an adolescent study in which we have developed a technique where we can extract the gut bacteria very quickly. We can put them in one capsule, then a second capsule, kind of like handmade chocolates. We've shown that the bacteria survive that, and we've shown good diversity in bacteria. Now, all of the other studies I've talked to you about haven't done that bit, so that bit we've got anchored. And we're about to begin treating obese teenagers with samples from very lean, fit donors. It'll be a double-blind RCT, and it's going to answer the question. It's going to be a thoroughly designed study and ask the question, can you transfer bacteria from those who are lean and healthy, and can that lead to appreciable weight loss? In mice, there's a 30% weight loss in body weight. I'm not sure we'll see that in humans, but in a year's time we can tell you the answer to that story. Thank you.